teaching you some neuroscience. So, why I'm doing this? I have an Academic Decathlon competition coming up very soon, and I want to know all of neuroscience, so I'm teaching all of you. So, as you can see up on screen, we have section 1 here. We're going to be going through 4 sections during this live stream, and those 4 sections are split up into the basic ideas of the cells in the neural system, and then we have the specific functions of those neurons and how they communicate with one another. And then we have all the systems within your body that have to do with neuroscience. And finally, we get into different technologies related to neuroscience. So we're going to go ahead and get started with section 1 right here. And as you can see, it's called Cells and the Anatomy of the Brain. So how the packet begins is it starts telling us how neuroscience is related to science as a whole. It specifically lists some things such as molecular biology, cell biology, physiology, chemistry, physics, psychology, cognitive science, computer science, and mathematics. These might be important to remember for a test for Academic Decathlon, such as a question like, which of the following fields is not often used in neuroscience? So remembering those basic ideas may be important. But what this main first paragraph mostly talks about is the idea of communication. Neuroscience is all about communication. You have cells that communicate in your body. That's how your brain works, is it has different cells within it that are constantly talking with one another. So we then get to, what are neurons? They're the main cell of the nervous system. And we want to understand them. So we know that they're all about communication, and we know that they can communicate across long distances, at least comparative to their size. So imagine a very microscopic neuron, like you can't see it, traveling all the way from, say, the end of your arm all the way up to your spinal cord or even to your brain. That's a pretty far distance for the cell. So these are some of the most fascinating cells in the body. So I also have a whiteboard here that I will be using throughout the stream to illustrate different things. And that's important because I'm going to be showing some different neurons on there very shortly. So, neuron. A neuron, another definition for it is nerve cell. It's a cell that has this name called a nerve. Now, there's one important aspect that you may be tested on or need to know about neurons, and that is they do not regenerate. They are set since your birth. You do not get any more neurons, except for very specific, like special types of cells, which we will get into later. But what's important to know here is they cannot regenerate. Okay, so neurons are a eukaryotic cell, which means they have what's known as a nucleus, which has DNA within it, and it can encode different instructions and make proteins. Then we need to know the different parts of the neuron. So this is where I'm going to start by illustrating a neuron here. So as you can see with this image here, we can see a sort of squiggly middle. Okay, let's just imagine this is our squiggly middle, okay? And this right here will have different certain cell bodies, such as the different proteins floating around your nucleus. So your nucleus is where all your DNA is stored. And this is called the soma, okay? Soma, if you literally translate it, it literally just means body. So this is the body of the cell. So then we also have different structures that can branch out of it. Okay, so let's say we draw these branches here. These different branches, they can be long or short, depending on the type of neuron, but these are known as dendrites. Now, what dendrites are different places where input comes into the soma. So, for example, you might have a signal coming at this little branch right here, and it'll travel all the way down to the soma, or the cell body. Now, this is different in different cases, but this is usually how it works. So, then there's another part. You may be seeing in the gut diagram, another part is labeled an axon. So, let's draw that here. So, we have this axon, okay? And what that does is it sends electrical signals to be sent to other neurons. So for example, this axon may stretch all the way down to some dendrite of another cell, of another soma. And at the point at which it attaches to the other cell, this is called the synapse. Okay, so this is your axon. This is your synapse. So, what the synapse is essentially where you're going to be putting all the neurotransmitters, which we can get into right now. So, how a neuron usually communicates, aka nerve cell, remember, is through these little things called neurotransmitters. Okay? And so, how they work is they move down the axon, and they move from your cell body all the way to the axon to the dendrites. Then those dendrites pick up the signal. And we'll be getting more in depth into this process of how the neurotransmitters are released and the different methods at which they activate the dendrites to activate the neuron. But all you need to know for now is that you have dendrites, soma, which is the body, the axon, and then other part to know is this part where the soma and the axon join is known as the axon hillock. Okay? And then the place right before the synapse at the very end of the axon is known as the terminal, the axon terminal. Okay, so we can go ahead and see some more important parts of this. Okay, so as you can see, the nice little diagram here was very nicely drawn for us as I drew on the whiteboard. And what's important to remember here is there are different structures of this. So the image you have on screen isn't what every neuron looks like. It's just a basic idea. And same on this whiteboard. So we're just using this as a model. But what we can say, if I were to go ahead and just erase all of this mess. Okay. So let us take a look at just the axon for now. Okay? So if we were to take a deeper look, I'm just going to put a little magnifying part here. We have our axon, okay? Around that axon, we have things called myelin sheaths, okay? So these myelin sheaths make up different parts the axon. Now, these cover the axon in order to insulate it and to carry the signal better. So, what we have here is, it says specifically that myelin are formed by glial cells, which we'll get into later, which insulate axons. They are similar to plastic or rubber insulation on a copper wire. This serves to speed up the conduction of the neuron's electrical signals that travel down the axon. Myelin insulates the axon in long sections with small gaps in the myelin that are called nodes of the reindeer. So, these little gaps that I illustrated here between the myelin sheaths, they're known as the nodes of the reindeer. Okay, so what does this all mean? How can I apply this, essentially? What the idea is that your nerve cells need this insulation in order to speed up the chemical process. Otherwise, it would be too slow for your body to communicate, and you probably wouldn't be able to think. So, what's also important about this is it noted glial cells earlier. What glial cells are the other types of cells in the nervous system. So, what they do is, or what a few types of them do, is they produce myelin to cover these axons. And what a question might come up is, it might ask you, what would happen if this type of glial cell were to stop being replicated in the body and you have less of that glial cell? Well, we just learned that glial cells formed myelin sheaths. So, what we could conclude from that is that the signals in your neurons would likely slow down and they wouldn't be able to transmit a signal, or at least as efficiently. Now, back to the nodes of the reindeer. What they do is they allow the electrical signals in from the outside of the cell, because all these electrical signals, they're really just carried by different movements of ions, which are cells, or not cells, atoms with a certain charge on them. So for example, you know the atom sodium, okay? Sodium is known as Na, and we know that atoms are made up of electrons, neutrons, and am I forgetting the name? Protons. Protons, neutrons, and electrons. Now, what happens when the number of electrons differs is if we have, say, one less electron, we would have sodium positive. That's an ion. It has a different amount of electrons than it does protons. So what this does for the axon is it allows a signal to be sent. So that's why we have these myelin sheaths, is to keep them insulated, keep the signal in there, inside of your axon. Okay, so we have a basic idea of how different neurons work. Okay. So, we have a different, one moment, I'm going to be right back. Okay, so just one more thing about these cells is that these dendrites are receiving the signal that's sent down the axon, not via the electrical pulse always, not always, but usually it's actually through what's known as the synaptic cleft. So this is a dendrite, okay? This one on the bottom is a dendrite. This is the synaptic cleft. And then this is your synapse, and this end part is your terminal. How this usually works, and we're going to get into this in section 2, is different ions trigger the release of neurotransmitters, which go into the dendrite and eventually activate the cell to activate a signal to be carried down the myelin. Okay, so we just learned all about the structure of a neuron, and we learned all about the passing of a signal and why that's used. So, now that we've learned that, we can learn more about the different structures that help neurons do what they need to do, which is communicate. So, if you have the Neuroscience Packet handy, you can go ahead and flip to page 8 and follow along. I forgot to mention that earlier. This is all through the Neuroscience Packet, which I should have put in the comments section as well, but alas I didn't, but that's okay. Let me know if you do want it, if you don't have it for some reason. But, okay, so let's move on. Let's see, is it working? Is it? Yes, it is. Okay. So, next up, we have just more names to know. So, what we know is that we have our axon here, okay? So imagine an axon that's coming up, okay? And then we have our dendrites, okay? And they're like little wires. So something important to know is that the cell that is giving the signal off to the next neuron, that is known as the presynaptic cell, or neuron, presynaptic neuron. And then the one that's receiving it via usually dendrites is known as the postsynaptic neuron. Okay? And what we also know is that the neurotransmitters that we spoke of earlier that go down through the axon terminal and through the axon really, or through the synapse, is packaged. Okay, so we have our little neurotransmitter. Let's say it's shaped kind of like this, where it has like a dot, and then a square, and then a triangle, okay? So these are the different proteins that might make it up. And this neurotransmitter doesn't absolutely work alone, because your body, being the wonderful creation it is, decided to try and destroy any possible invaders so that you could be protected against things like viruses. Now, this doesn't exactly help our bodies always, though, because we could have a protein that's completely harmless and won't hurt us at all. In fact, it'll help us. But your body has these enzymes that are floating around that like to, let's just say, bring a chainsaw and they like to kind of just destroy those proteins. So we need a solution for this. That solution is a vesicle. So we have this little fatty casing that surrounds our nice neurotransmitter protein. So these vesicles are used to transmit signals across the synapse or the synaptic cleft. Okay, so now that that's good, we can move on to what's next. Okay, So next, yes, okay. So we have different types of neurons. So as I said earlier, we might have a what we think is a typical neuron is the soma, and then we have dendrites that stick out of it. And then we have our axon, okay? And then we have our little axon terminal. So, firstly, let's do one more piece of terminology, and that is the dendritic arbor. So the dendritic arbor is just the whole collection of all these dendrites and the different branches that come out of them. So all it's saying is your dendrites from your cell when they say dendritic arbor. So don't be confused by that. And then we get into the different types of neurons. So I'm going to go ahead and just show you this image here on screen. You should see it now. It's 3 different types of neurons. We have our motor neuron, which is what we usually think of as the typical neuron, which has the dendrites and the soma and the axon and the synapse at the end. Then we have interneurons, which are like just a lot more messy, honestly. We have this cell body, and then we just have all sorts of things just coming out both sides here. Usually it's that one side will give off a signal, so like this signal might go this way, but they're really branched all over the place because what their function is, and this is another part of neuroscience, is their shape often is based on their function. So if a cell, for example, if a neuron, an interneuron, if its job is, which it is, its job, is to relay signals and send them out to different neurons, this would be the perfect structure. Because then what it can do is it can take in signals from all over the place and then relay them back all over the place to multiple different other neurons. These are especially useful in the brain because we're constantly relaying signals and modifying them and strengthening them, so we need to be able to have these almost like a mediator. If you're playing a game of telephone, imagine Bobby A is standing right here, Bobby B is standing right here, and Bobby C is standing right here. If Bobby A wants to talk to Bobby C without directly talking to him, he can talk to Bobby B. So I could go like, neuroscience is cool. And then Bobby B will be like, neuroscience is cool. And then Bobby C will be like, oh, yeah, neuroscience is cool. I agree. I agree. Yay! So that's the idea of an interneuron. Okay, so then we have a motor neuron. So think about your muscles, okay, or just point your finger for me. If I were to think, okay, I'm thinking really hard, and now I suddenly think I want to move my finger and bring it down into my hand. So I'm going to do that, okay, and then I'm going to bring it back up, bring it back down. Notice, there aren't many signals just going back and forth between one another. It's going straight from my brain to my finger. I tell my finger to move, and it moves. So this means that the neuron often has to talk going towards it. So that's why we have this structure of an axon, a long axon, being able to talk to the different muscles where I want it to move, and then we have the dendrites on one other side so that we can talk even more, so that it can relay the signal all the way from my brain. And these can stretch a pretty far distance, if I can remember, yes, because it has such a long axon. Okay, so okay. Next, we have sensory neurons. Okay, so I'm just going to read this section and then I can explain some of it. Sensory neurons vary widely and are very different from motor neurons, as their role is to bring sensory input from the body to the brain. So note, this means if you get touched in the hand, for example, it's bringing that sensation all the way through your arm, all the way up your neck, all the way to your brain. Okay, from motor neurons, as their role is to bring sensory input from the body to the brain. Some sensory neurons have dendrites at both ends connected to a long axon with a cell body on a branch in the middle. Interneurons work to connect other neurons within the nervous system and are often quite small without long axons or large complex dendritic arbors. In this way, the structure of a neuron is directly related to its function. Okay, so that's the basic idea behind sensory neurons is since you're relaying just a sense the entire time, you can have these dendrites on both sides, or not dendrites on both sides, but we can have axons on both sides that carry the signal so that it goes all the way, and we have our cell body so that it goes all the way to our preferred destination. We'll also later learn that this doesn't absolutely have to go to the brain. Have you ever been to the doctor and they hit your knee? Well, that is actually just a signal going straight to your spinal cord and then right back to your knee. So that's why these structures, even though we have a basic idea of, oh yeah, that's what a sensory neuron looks like, or oh, that's an interneuron, they can all vary because they all can have slightly different functions in the body. Okay. Next up is white matter and gray matter. Okay, so this part is pretty self-explanatory. Mostly what it's talking about is how we have two different sections of all neurons, and that is gray and white matter. Gray matter is usually the bodies, the cell bodies, the soma of your neurons, and then we have the white matter, which is all the axons and such. What's important about these is we can then tell, based on if it's white matter and gray matter, where the signals are being relayed in the brain. So, for example, we know that on the inside there's a lot of white matter because that's where a lot of the axons are stretching to one another. And then the outside of the brain is usually a gray or darkish tan. If you were to go ahead and just get yourself a brain, you'll be able to see this. So we know that white matter is the axons usually because myelin itself has a white pigment. And it's a fatty tissue that can surround it, and that's basically what you have to know. Let's see. So we know that the inside, perfectly demonstrated on this little figure 6 here. The white part is your inside, your axon, and the outside is the soma. This could make a good test question too, because it could ask, if you cut open a brain, what will you see on the inside? And it will say, like, option A, DNA, trying to trick you about the cell bodies, or the soma. Option B, the spinal fluid, I don't know, or cerebrospinal fluid. Or C, myelin sheaths. And it might not even say that. It might say something like the material made from glial cells. It may try to trick you like that, but just keep on your head that the white matter is the myelin sheaths on the axons. Okay. And we finally get to what are glia. Okay, so this is a fun part because I get to explain a story. So, we have two different people that are working in an office one day. We have one man, and he wears a nice little top hat, And his name is Eligio. Okay, everyone loves Eligio. Everyone knows Eligio because he's working on the parts of the brain that are really recognized. You know, he's working with your brain and your spinal cord, probably the most important parts of your brain. And this is like a little factory in your brain. So you have Eligio here, and he's all happy because he gets to work in the central factory, okay? In the central factory, yep, I spelled it right, he gets to, with his special tools, okay, he gets to form these blobs, okay? And these blobs, they are myelin. They then get to go and be shipped all the way to your brain or to your spinal cord. And the myelin gets to go there. But then unfortunately, we have another. We have who is known as Schwann. Okay. Schwann, I mean, you can tell by just the name. Schwann is a bit of an oddity. He doesn't really interact with the others much. He's kind of, you know how in your vision you can look at something and it's in the central part of your vision, but then you also have this peripheral part, which is important, but we don't always really notice it. So Schwann is part of what is known as the peripheral factory. Okay? And in the peripheral factory, there is literally all the rest of your body. It doesn't get to interact with the brain or the spinal cord. No, Schwann has to sit in an office all day, making his myelin, okay, at a desk, alone, in a cubicle, okay? So Schwann sitting, making his myelin, all sad, because nobody really notices him. He's really just a peripheral. So, each of these people are very important, though. You may know that Schwann, he's alone, and Eligio is well known, but in the end, they both make myelin. So, where this analogy went, I think it may have went too far. But Eligio is what is known as the oligodendrocyte, which, notice the root cyte, which means cell, and then oligodendro, that part just means it's for making myelin in the central nervous system. So your oligodendrocytes are the myelin makers in the central nervous system. And then Schwann cells, okay, they're just known as Schwann cells. See, they even get a boring name. I feel so bad for them. Okay, well, I feel bad for the guy who Schwann cells were named after. I'm sorry, Schwann, whoever you are. This is all just a joke. It's just so they can remember that it's the central, and I didn't get there quite yet, nervous system. Okay, so the central nervous system, this leads us perfectly into the central nervous system because it's what makes up our brain and our spinal cord, as I said earlier. That is the parts of the central nervous system, so if you ever see this referred to in this way, it's that. And then your peripheral factory is actually really just your peripheral, say it with me, nervous system. Okay. Nervous system. Okay. So Schwann cells then get to create all the myelin for, like, the neurons in your hands and your body, other than your brain and spinal cord, your legs, okay? So this is all peripheral. It's not central, because remember, your brain and your spinal cord are all central to your body. That looks like a banana person. So, yeah, next time you ever are talking to someone who knows neuroscience well, just go and say to them, did you know swan cells are the banana person of the body? Yeah, that'll be fun. Okay, so they're part of the peripheral nervous system. They're making myelin, which is the fatty tissue which goes around the axons, and that's their job. Okay, so next, I probably shouldn't have gone on a big old rant about Schwann cells and oligodendrocytes, but I really wanted you to remember which each of them were, so hopefully that helped. Okay, finally we get to the star of the show. We get astrocytes, named after their star shape. And all glial cells actually used to be thought of like this. They used to be thought of as just these passive cells which glue together the brain, but really, they're much more important. We learn that astrocytes, and remember, when we're looking at cells, we can look at it through the perspective now of morphology, okay? Okay, which, remember, is the shape / structure, okay, of a neuron. And notice, like, if you're struggling with that word, morphology, just know it's morph, like you're morphing into something else. Imagine that superpower. That's how you'll remember it's the shape / structure. But we know that astrocytes have this star shape, okay? So we have this center, and then we have all these little branches that come off of it, okay? So we know that the shape and structure is usually based on function. So what we can remember from this is that astrocytes, they do a few things. They form the blood-brain barrier, which we'll get into. Let's just list them. They also regulate the chemical makeup of the extracellular fluid in the space between cells of the nervous system. They can also remove excess signaling molecules to allow proper balance of ions. They can react to tissue damage, and, as I said earlier, they form the blood-brain barrier. Okay, so these are all about regulation. So not only do they form the blood-brain barrier, but they also can regulate the fluid. Now, it's in the extracellular, so that means it's outside of your neurons. The fluid is the extracellular fluid, which means outside of the cell. So then they also maintain proper balance of ions. Yeah, so this is kind of grouped with the certain, like, concentration of ions. So imagine we have, like, a neuron here. Okay, I'm just going to put a little actually, I'm going to do an interneuron, because this is usually what the brain is. Okay, and so we have our little astrocyte here that can wrap around this neuron so that it can protect it from any diseases. Now, we need to note that the blood-brain barrier is only present inside your brain, and I believe the spinal cord. Let me check. Entering the brain. Yeah, I think it's just the brain. But the idea behind it is that they're going to wrap around the brain. Now, this could cause problems, though, because if we're trying to get medication to the brain, these astrocytes are going to stop us from getting that medicine in, because we have our bloodstream here that goes through the brain, and we're trying to get the molecules in, but the astrocytes won't let us. And then also, what astrocytes can do, I imagine with all its branches, like, this is how I remember this, is it has, like, almost like its octopus legs. It's like going around and healing all the neurons, trying to fix the tissue as well, so that's the reacting to tissue damage. Okay, so those are astrocytes, and that's the basic idea behind them. Also note, the blood-brain barrier, it's not present in your peripheral nervous system whatsoever, so that's why it's easier to get medication to, like, numb your body than to, like, completely just, like, numb your brain or something. I don't know. But to be able to fix problems in the brain, it's much harder because of these astrocytes. So we kind of have a built-in defense mechanism, which also kind of defends against ourselves in a way. I guess our bodies didn't really account for the fact that we would get so smart and make medicine. So we have our astrocytes, which are seen in figure 7. Okay, and finally, we get to the microglia, okay? So these are, as their name suggests, very small, okay? Probably the smallest of these. So they just make up different, like, branches almost. Just imagine them connecting to one another, almost like the image shown here. And the branches are almost like blood streams, I believe. And what they do, they're like the immune system of your brain. So if you ever have some mean neurons, you ever have some neurons that are just like not having a good day, okay? They're just being mean to everyone and like hurting everyone. They're like, ooh, I don't really like you. I'm gonna hurt you. And then what the thing will do, what the microglia will do, is it just eliminates them. It throws them in the trash. And if the neurons are being fine, but they are dead, then the microglia will come and clean them up and recycle them from supplies. So that's important to know. And then, let's see, what else? It can be a defense for infection. And then we also know, though, that it can cause neurodegenerative diseases such as Alzheimer's. So we have our microglia. They may seem harmless, but they can, because they can destroy neurons, they certainly can hurt the brain and cause things like Alzheimer's. But that is the role of microglia. Okay. We are now on to the anatomy of the central nervous system. So, we're not going to be looking at the peripheral nervous system for a while now, but we will be looking at how everything is connected in the central nervous system. So remember, that's the brain and your spinal cord. Okay, so we can first start with the meninges. Now, the reason we want to learn this structure is because structures, remember, morphology, are very often related to the function of a system. So, starting with the meninges, okay, we have the different... Oh, I hear a lizard. Hello there. So, we have different parts of your skull that are protecting your brain. That's where the meninges come from. So, most of these are actually Latin. So, I am going to go ahead and get my marker out here. So, we have our skull. I'm going to also scroll down to the image so you can see it better. Yeah, that's good enough. Okay, so imagine we're looking at the top part of our skull, and I know you have the image here, but this will help to just like as I illustrated, I can explain it. So we have the bone, okay, and then we have what's known as the epidural, so epidural space, okay. Now, this is the part that's connecting the dura mater, and that's spelled with one T, so dura mater, I guess, I don't know. But we have like stuff flowing through here. So that epidural space is used to absorb shock. So absorb shock. So we're absorbing the shock from, say, I hit my head right now. Oh, no, I got epidural space to cover me. I'm not going to lose any neurons, don't worry. Well, I might, but this probably isn't a good idea. So what we have in the next layer is the dura mater. So it's to protect it once again. And so what we also use epidural space for, since the rest of the parts of the meninges, which protect the skull, are so tough, we just kind of, if we ever want to inject medication, we just like do a needle through this bone, rather than having to go all the way down to the brains and neurons themselves. We have this, what's known as cerebrospinal fluid, which flows through the subarachnoid space, which is 3 spaces down. So we have our subdural space, we have our arachnoid matter, and then we have our subarachnoid space, okay? So what we can know is that the dura mater is very tough. Dura means tough, okay, in Latin, and then mater means mother. So that literally means tough mother, which means this is like a tough mother that's protecting your brain at all times. So then we also have the subdural. This means less than tough, so it's right below the tough mater. And then we have our arachnoid matter. Arachnoid matter. Notice that it's matter with one T again. So this is your arachnoid mother. This is it's just the space underneath it. It doesn't speak too much about it. But then we get to the fun part, your subarachnoid membrane, or space. Your arachnoid membrane is the combination of your arachnoid mater and your subarachnoid space. Okay, so it's made up of these stringy components, which, if you think about it, this makes sense, because it's meant to like carry pool, like it's almost like a pool that's going through the stringy matter. There may be pressure that needs to be released from the subdural space. Okay, so we have our subdural that might build up in pressure over time, and so it needs to release its pressure into this more open and stringy arachnoid matter, and then into the subarachnoid space. And this is also what is filled with cerebrospinal fluid. So since it's so open, it's going to be filled with that fluid. And then finally, we have our pia mater, which just means gentle mother. So pia mater. Okay, so the reason it's called gentle mother is because it has to perfectly align with the grooves and bumps on your brain. Now, you may be asking, Bobby, is there a specific name to the bumps and grooves on your brain? And I would say, yes, even more for you to be tested on. So, we have specific names for the bumps and grooves and such on your brain. And if I can scroll down, maybe it'll let me scroll down so you can see. Okay, this is just going to be annoying, huh? Okay, you guys can see the scrolling. I can't see the scrolling. So, you guys will be able to see it, but that's okay. So, we have the bumps and grooves. Okay, so like this is your surface of your brain, okay? And then you have these little bumps and then are sticking out like little mountains. It's very peaceful. Could be Bob Ross. Just paint and draw all day. Okay, so we have our bumps and then we also have our grooves. So these are our grooves and then these are our bumps. And they're also, if I were to take a look here, because I can't see on mine. Okay, so we have our grooves. They also say they're called folds because, I mean, that makes sense, right? They're folding inward. Then we have our bumps. Now, we have our gyri, which are bumps, and then we have our sulci. Okay, so our, and they're also gyrus, and sulcus. Okay, so how I remember gyrus and sulcus is sulcus, it sounds like you're kind of sulking, like, you're sulking around, you're kind of down. You get these folds in your skin, you get bags under your eyes. That's folds and grooves. So you get folds on your eyes when you're sulking, so sulcus. And then you have gyrus, which are like, imagine like a, just the other one, essentially. Gyrus, you can remember it's the bumps on your brain. So, then, we know those names. And, we can go ahead and erase that. So, back to pia mater, we remember that pia mater is going to go over those gyri and sulci, and it's going to form a perfect barrier so that it can keep it insulated within the brain. Now I'm going to go fix my iPad so that it will actually start working. Might look weird for a second, but that's okay. Okay, perfect. So, now both me and you can see the iPad, which is wonderful. Okay. Next up, on the inside of all that meningy mess, the meningy mess. Okay, so we have three main parts of our brain. Okay. Okay, so we have the cerebrum, the cerebellum, and the brainstem. Okay? Cerebrum, cerebellum, and the brainstem. Okay, so now what we're going to do is we're going to label each of these parts according to the different things that are in them. So let's start with the cerebrum, okay? So the cerebrum is pretty much everything except for the brainstem and cerebellum. It's what you think of when you think of the brain, okay? And it does most of the work. So we first then get to, let's see. Cerebral cortex, okay. So we have two main parts of the cerebrum. We have, now I don't have a 3D whiteboard unfortunately, but if I did, I would be able to show you that there's an outer layer and an inner layer of the brain. You can imagine that. So on the outer layer, that's called the cerebral cortex. Okay, and we're going to talk a little bit about that. So on the outside, in the cerebral cortex, this isn't really to scale. This isn't actually how it looks. It's probably more like this. You have these different lobes, okay? So this right here is your frontal lobe. This is, sorry. This is your occipital lobe. Temporal. Why am I forgetting the top one? No, not again. Parietal, yes. Okay. So we have our parietal lobe. Okay. So each of these lobes have different functions. So, for example, the frontal lobe usually has to do with your motor movements and also your decision making, and also it does have some stored memory, but not, it doesn't really do as much as the temporal lobe does. It's basically the control center of your brain. If you imagine the movie Inside Out, like the frontal lobe is probably where all of that is happening. It's also associated with, as I said, decision making, but also like your like almost like survival instincts, I could imagine, but like more like intuition. It's a better idea. It's your intuition, and the thing is, teenagers, including myself, do not have a fully developed frontal lobe yet. So that's why we are so stupid. Anyways, the parietal lobe is where different things such as your sensory systems are, your I wish I could scroll. I'm gonna have to fix this again. But let me just okay, so the frontal lobe, it's your decision making and your social behavior too, as I said. And then we also have thinking, feeling, cognition, as I said. Prefrontal cortex. Okay, so all of this in your frontal lobe, it's just also known as your prefrontal cortex. Then the parietal lobe, like I said, it helps with sense of touch from your body, skin, organs, temperature, pain, and body position. So if you're thinking about like spatial relationships as to where you are, then like that's where your parietal lobe is coming in. If you can see an object, this is where some of your vision processing, which we'll learn about later, is actually thought of. If you can see an object in 3D, that's your parietal lobe processing that. So, then we have the occipital lobe, which, visual information, that's literally it. You have an entire lobe of your brain entirely dedicated to just seeing. How lucky are you that, like, your entire occipital lobe just dedicated to seeing? So, that's important. And then, the thing is, we're going to learn later that, for example, say you're blind. You have no need for this occipital lobe anymore. That's at least for visual processing. What's fun about your brain is it adapts, especially early on. So if you're blind early on, your brain is going to develop to extend to that area to be able to do other functions better. So, for example, you might have a heightened sense of touch or hearing when you don't have a sense of sight, because your brain is then using that segment in order to enhance those abilities. So, like, you may know that blind people, they might have better senses in other senses. This is why. It's known as neuroplasticity, where neurons can adapt and change based on their function. Okay, then we have the temporal lobe, which is mostly just auditory. So you have your speech processing, music, memory, and object recognition. And several important regions can be found in the temporal lobe under the cortex on the surface. Okay, yes. So then we have the cerebellum, which we're going to get into later as well in section 3, I believe. But it's pretty much what you need to know is it's where your movement processing is. So you might learn how to ride a bike, and you know how you almost never forget how to ride a bike. That's because of your cerebellum and all the processes. Now, an important fact that it likes to note in the packet is that there are twice as many neurons in the cerebellum as there are in your prefrontal cortex, or not prefrontal cortex, just in your cerebral cortex as a whole. And then we finally get into the brain stem. But not yet, actually. We actually get to subcortical structures. So, if we go ahead here, and draw this out, we can draw our basal ganglia, that's not what it looks like. That's a pancreas. Okay, that's better. Then we have all these little things here. And then we have our hippocampus. And then we have our amygdala. And then we have our thalamus. Okay, perfect. So, let's start by discussing what these are called. These are called the subcortical structures. So, sub meaning below, cortical, and then structures. So they're below the outer portion of the brain. From there, we have, let's start with the amygdala, as it says. It's right next to the temporal lobe, or it is in the temporal lobe, but it's like in the very middle of it, where, now, pop quiz, where do you think, is it gray matter or white matter that most of the subcortical structures are surrounded by? I'll give you until 3, 2, 1. Okay, it's more likely white matter because it's on the inside as opposed to the outside. Because remember, the outside of the brain is your gray matter, and then your white matter is your myelin sheaths, if you remember from the axons. And so the amygdala, it's where the temporal lobe is. And amygdala sounds kind of like almond, right? And it kind of looks like an almond, so we can like kind of imagine an almond here. And that's where it comes from. It's Greek for amygdala. And it's a part of the limbic system. So I'll just go ahead and list some of the things it regulates. It regulates fear, threat, stress, reward, and arousal. So these are emotions, and it can also focus on emotional memory. So it can be found in... Now, this is what's fun, is not all of the subcortical structures are found in white matter. This amygdala, is actually found in the gray matter, or nuclei. Now, I forgot to mention this name earlier. Gray matter is also known as nuclei. Can you guess why? It's because the soma bodies are the gray matter, and they have nuclei inside of them. So we just go ahead and call them nuclei. Almost like how later on it's going to talk about hydrogen ions, and how they are one proton and no electrons, and some neutrons. That's why we often just call hydrogen ions just protons. So, since these have nuclei in them, we just call them nuclei. Okay, so then let's move on to the hippocampus. Oh, not again. Okay, so the hippocampus is important for, let me just read back up again on it. It's good for two main things: spatial navigation, and then we have long-term memory. Okay. So we know that it's a part of the limbic system as well. So we now have two members of the limbic system, which processes emotion, remember? So we have the amygdala, and then we also have the hippocampus. So we have amygdala right here, and then its neighbor, the hippocampus. The hippocampus, it's derived from the mythological version of the seahorse. So that's because it kind of looks like a seahorse, even though it doesn't. I don't know what they were thinking. This is a seahorse now, apparently. This is what a seahorse looks like. I mean, if anything, it looks more like a shrimp. I don't know about a seahorse, but whatever. You be you, mythological creatures. Then, what's important about the hippocampus is that it processes long-term memory. So this is known as memory consolidation, and we are able to store our memories for long-term use in the hippocampus. Okay. I am going to go fix my thing again, because it seems to keep malfunctioning. Okay, so back to each of the different structures. Back to the hippocampus as well. We have our spatial navigation and our long-term memory. It mentions the words memory consolidation. So this is what consolidation is it's choosing where it's going to go. So for example, in the brain you might have the frontal lobe, and then you have the parietal and temporal, so forth. But the brain needs to decide where all these memories are going to go. Is this like an auditory memory? Like if you're listening to a song, you're going to remember that song in your temporal lobe, versus if it's like an emotionally attached idea or memory, it's going to go to your frontal lobe. So what consolidation does is it takes it from one place. So imagine this little square here inside of this circle is your memory, okay? The circle around it is going to decide, hey, is it going to go this way? Is it going to go this way? Or is it going to go this way? It's consolidating. So then we can go ahead and move on to the next part. My goodness, it just does not want to help me today. Okay. So next up is our thalamus. So if I were to scroll down here for you. Okay. We have the thalamus, which is in the center. So I'm going to erase all of this. Set this down. Redraw the brain since it's pretty messy. And then what I'm going to try and do is I'm going to fix this because it is just not wanting to cooperate right now. Okay. So okay, hopefully this will help, but it may disconnect again, which I will be annoyed. That's okay. So, back to the thalamus. Okay, so we have our brain. Okay. This is our cerebellum, and then this is our brainstem. I know, wonderful brain. So, we have in the center here our thalamus, and we can also imagine our brainstem going up to it because that's going to be important, because the thalamus is your relay station. So imagine a bunch of little buttons and switches that are in the thalamus, because that's what it does. It's relaying all of the information that comes from your body through neurons all the way up through your brainstem. It's going to relay all those messages. Now, the main notable exception to this is going to be your smell. So, you have your couple eyes right here. And then you have your nose. Okay. So, this is your face, and this is your brain. Okay, so your nose is kind of a special case because it just goes right past the thalamus and just kind of ignores it, because like it doesn't need no thalamus. Like it can just smell. Like, so that's the idea, is smell is the only exception to this rule. Otherwise, the thalamus, it is the relay station. So then we also know that the thalamus plays a role in consciousness. So consciousness. We have emotional behavior. We have motor control and cognition. So it's kind of like the first part of your prefrontal cortex. Because remember, your prefrontal cortex here, it processes what? Emotion, thought processes, consciousness, your logical thinking, motors, skills, and cognition. So it's like your prefrontal cortex junior. Then we get into the hypothalamus and its neighbors with the pituitary gland. So if we go back up here, we don't get an image of those yet, but we do. Where is the image of it? Oh, so hypothalamus, it's right below. It's like going to be around there. And then we have right next to it the pituitary gland. It doesn't have a diagram, unfortunately, but if you can imagine, we have the two parts right there and there. Hypothalamus and pituitary gland, which they are important for hormones. Now, here's the thing. Our bodies run on not just, oh, I eat something and then stuff happens, for no reason. No, our body is regulated by hormones. And these aren't just hormones that just happen, like people say, like, oh, you're going through puberty, you're going to get a bunch of hormones. No, hormones are used all throughout the body, all stages of life, because they're just a signaling molecule. They often get a different name than what they actually are. So, hormones, they're important. Like, for example, in my body, I'm a diabetic, so I do not have a working pancreas entirely. And what that does to me is it stops me from being able to produce the hormone insulin. And insulin allows me to convert sugar into energy. And it needs, in order for that to happen, it needs that hormone in order to convert it. So hormones are used all the time in the body in order to just regulate systems. Okay, so the idea behind this also is homeostasis. Okay, this is going to be a very important word. Stasis. Okay, so what this means is it's keeping a balanced state. Usually, this balanced state is created via two contrasting systems. And how this works in relation to hormones is there's usually, let's see, okay, so both of these are part of this releasing of hormones, okay? And the pituitary gland is releasing hormones, but reason why it's part of the nervous system is because it's attached to parts in the brain that can then send out the signaling molecules to, say, your bloodstream. And then we have parts of the endocrine system, which are pieces that aren't part of the nervous system but also regulate hormones. So, for example, your hypothalamus might activate parts of the endocrine system in order to do things for homeostasis, which is things like regulate body temperature. We have blood pressure, hunger, thirst, sleep, and wakefulness. All of this, it's used for survival. And so we actually, and this is a side note that isn't absolutely in the packet, but something important to know, is that part of what hormones are is they're specifically slow acting. So they they don't just all release at once into your body, and you aren't supposed to have just a bunch of hormones rush into your body like that. You have these two contrasting systems of hormones, and then we have steroids, which steroids can just go directly into a cell. So imagine we have a cell here, okay? A nice wonky cell. And here's your DNA and your nucleus. Okay, so hormones, let's indicate it by a triangle, and then steroids, let's indicate it by a couple hexagons put together, because this is what they actually look like, kind of. What steroids can do is they can go straight through the fatty tissue in the cell, and it can attach to different things in the cell, immediately affecting things. Then hormones, on the other hand, they have to go through channels in order to get in, or activate receptors on the surface of the cell, which we're going to learn more about in section 2 of the packet. So, that's the main idea of what you have to know about the pituitary gland and the hypothalamus. It's all about homeostasis and hormones. Okay. So finally, we get to the basal ganglia, which are gonna be part. Let's go ahead and just set that down real quick. We have our basal ganglia, which look kind of like, honestly, they look more like a seahorse than the hypothalamus did. Or, I mean, sorry. They looked more like a seahorse than the other one did. So, let's see real quick. My iPad, once again, is not working. So, okay. We have our hippocampus. Okay. Okay, and then we have our thalamus and our hypothalamus and pituitary gland, and then we have our amygdala. Okay. Okay. Basal ganglia. Okay. So what basal ganglia do are they're a bunch of nuclei. Okay. So this means gray matter. So it's on the outsides more. That are collected here in order to communicate signals, and they're almost like a control center. So if you were to go into your phone and like click the flashlight, for example, it's as if the basal ganglia are telling your flashlight, hey, it's okay to turn on. I'm the control center. I can tell you to do that. But what happens with the basal ganglia is they can be misregulated, and this can cause things like Parkinson's disease. So imagine this. You have your control center in your phone, but then you also have this side app. Now, the side app you bought is actually a virus, okay? And we don't like that virus. But your control center in your phone might say, hey, only I can turn on the flashlight, not this virus, okay? But if your control center were to suddenly stop working, then your flashlight might randomly start turning on and off. and it might, well, damage your phone. So in a similar way, our bodies need the basal ganglia to be able to say, hey, no, you can turn on or you can turn off, you're good or you're not good, to move, okay? This is movements, not turning on or off our eyes or something. This is all about movement, and so if they become misregulated, we can have things like Parkinson's disease, which can cause little twitches in your body when it's supposed to not be moving, or even you might have trouble controlling your body. Okay, so the main parts of the basal ganglia we will get into in section 3, I believe, or maybe it's section 4. But section 3, it talks about the caudate nucleus, the putamen, and the globus pallidus, and the subthalamic nucleus, and the substantia nigra, which is interesting because what the basal ganglia do is they actually interact with a part, let me double check this, yes, with the midbrain. So you have your basal ganglia, but then remember we have our thalamus here, and then we also have all the other parts of our brainstem, which we're about to get into. And the next section to go into is, so the thalamus is here. It's not technically a part of your brainstem. It's technically a part of your subcortical structures, or subcortical. The basal ganglia can attach to things like your substantia nigra and your, what was the other part? Subthalamic nucleus. So, subthalamic, remember, subthalamic. You can remember this because it's right below the thalamus. So your subthalamic nucleus, and you also have your substantia nigra. Okay. Those two parts, part of the midbrain, and they communicate on and off with your basal ganglia. Okay. So next up, if I were to fix this again. Okay. Now I can see it. Okay. So now we get to explain the midbrain, or the spinal cord. Okay. So I'm going to sit down to explain this one because there's not much to draw other than the diagram you can see. So here we have the idea that we have, the analogy it gives us is this scoop, this ice cream cone. You have 3 different scoops, okay? You have the midbrain, you have the pons, and then you have the medulla oblongata. You'll probably only need to know that's called the medulla, but the technical name is medulla oblongata. So we need to know the functions of each of these. So I'm going to go ahead and just explain each of them. So the midbrain is the topmost part and contains nuclei that help regulate eye movements and the pupillary light reflex, which automatically constricts the pupil in response to light. The midbrain also contains regions that help integrate visual information from the eyes with auditory information from the inner ear into a cohesive map of the environment. So this just means it's mostly visual and auditory stuff that's regulated in this part. So if you are, for example, in a very dark environment, what this pupil eye reflex does is you're going to be like looking in the dark. Your pupils are going to be very dilated, which means they're going to be very open. You can see a little bit in the dark because of that widened pupil. And then once the lights turn on, it doesn't want too much light flooding into your eyes, so it constricts the eyes. And then we have it's integrating it with the information with your ears and eyes, so that you know, hey, I hear a trash truck over there. I can not see it, but it helps me understand, hey, where is everything related to where sounds are happening? Okay, then we have the pons, which come from a Latin word meaning bridge, okay? So you can remember this because it's literally a bridge connecting the medulla, which is on the bottom, and the midbrain, which is on the top. And then above the midbrain, obviously, is the thalamus. So we have the pons, bridge, and it's mostly used for coordinating facial movements. So, and then hearing, and then balance. So if I were to go ahead and stand on one leg right here, I am using my pons. I also imagine it like pawns sound like a pawn from chess. So pawns, they're used in chess, if I'm not mistaken, for they are very useful, and you want to keep them in balance, right, with your opponent. So what you can think of is pawns is keeping you balanced. It's also keeping you coordinating facial movements. So it can coordinate attacks in pawns too. You can create like entire structures. I'm not the best at chess, but this is just the idea behind it. Hopefully it'll help you remember, at least, because it'll help me remember. And then it also helps with hearing. So if you think about balance too, balance is often connected with hearing because we have these structures in our inner ear which allow us to understand where our environment is. Okay, then we get down to the medulla, which is referred to as the medulla oblongata because it is long and rectangular, as I said earlier. And it's used for touch sensations from the face, swallowing food, vomiting, blood pressure, heart rate, breathing, and other functions. So this is all like reflexes. So like if you are getting really stressed out, the medulla might regulate your heart rate, help you breathe better. If you need to swallow food, you don't really think about it. It just kind of happens, right? So that's that. Then we also can think about this in relation to gray matter and white matter again. The brain stem has gray and white matter because it needs to relay signals and receive them. So we have lots of gray, which is the soma or cell bodies, and then white matter, which is the axons and the myelin. Okay. Next up, we have the spinal cord. Okay. So, first thing to note is that the white matter is on the outside of the spinal cord and the gray matter is on the inside this time. So this is the opposite of your brain, where all the cell bodies is on the outside. It's in your cerebral cortex usually, so the outer part, as opposed to just your cerebrum as a whole. And so you're going to have that gray matter on the inside this time for your spinal cord, and the white myelin sheath axons on the outside. So from there we also have it's useful for spinal reflexes. So once again, I said, like, if your doctor hits you on the knee with a hammer, then you're going to see that. And then we have different sections of the spinal cord. So as you can see here in this diagram, you have your cervical spinal cord, which is like up here, okay, all like up to your neck, up till where it ends, all the way to about the middle part where your chest is, which is known as the thoracic. And then you have lumbar and sacral. So how I remember these is your cervical spinal cord. I see that C is the earliest letter in the alphabet, so cervical is in the top. Your thoracic, I think of like I don't know, I think of thorax for some reason. So that's probably where it's named after, is it's this section. And then the lumbar, you can remember that because if you have a chair, there might be an advertisement saying lumbar support. That's going to be just your lower back right there. And if you think about it, most people like often look for the most support in their lower back because that's where it hurts a lot. And then your sacral spinal cord, just think of I don't know, sacral. It's the bottom part. Honestly, I don't know what connection to make with it. It's your sac. Maybe you can think of it as the bottom of you, so it's where you're carrying a sack. And then thoracic is just the middle. You can think of, then it's process of elimination. So, no. Oh, not again. Okay. You can see it. I can't see it. So, I need to fix it again. Okay. Okay. So, we finally get to... Okay, so then finally what you'll need to know is that there are vertebrae. So that means there are little sections of bone. Rather than being completely covered like your brain, you have these little sections of bone, vertebrae. And in between here is where different nerves can go out and send information to nerves in your body. So for example, each part of your spinal cord, it communicates with a different part of your body. So the cervical spinal cord, it innervates, which means it's connecting with, just innervate. Imagine like something connecting. I think of the word innervate already. But it's like your inner working, your intertwining, innervating. So then, the cervical, it controls your arms, neck, and shoulders. Okay, so then we have the thoracic spinal cord, which carries information. So chest and torso, as I said earlier. And then lumbar, hips and fronts of legs. And then sacral is the buttocks and the back of legs, as well as the nerves. Okay, so then each section of the spinal cord also controls all the nerves that run out of the little sections between vertebrae. Finally, we get to the ventricular system, which is probably the most complex part of this, just because it's kind of hard to keep track of. So we're going to go ahead and draw ourselves a diagram, because otherwise we will not be able to keep track. Okay, so I'm going to go ahead and just draw it based on what I see here. And then, yeah. Okay. And, yeah, the diagram just isn't very well put together, so I'm going to try and rework it for you guys. Okay, so this is our brain. And we have our lateral ventricles here, okay? So, imagine two of them, okay? Or not really two of them, but imagine it in 3D. So, you have it, they're like two little sacs. So you have your right lateral ventricle and your left lateral ventricle, which are on each side. So, like if you're facing me, that's gonna be your right lateral ventricle, and well, no, because you're looking at the screen. So actually, yeah, this is your right, and this is your left. Obviously, you can figure it out. It's just your right and left are where your lateral ventricles are. And then with those are each connected to what is the right and left thalamus. So you have your thalamus in the middle here, and then you have your lateral ventricles, which are connected. I'm just going to draw it as one, though, so that it's simpler. So imagine you only have one ventricle, and then you also have the courteous plexus here. Yes. You have your courteous plexus, and then you have your interventricular foramen. Okay, so here we have the different parts which can go into the brainstem and so forth, and this actually connects to there. Okay, so let's label each of these parts. This is your lateral ventricles. And then you have your courteous plexus. And then you have your third ventricle. And then I'm also going to draw the canals right now so that I can show you what it is later on. Okay. Then we also have the interventricular foramen. Okay. So with each of these parts, each of them have a different structure, and then we also have, okay, so here's where our spinal cord is. I already showed it, kind of. But there's also, like, kind of two parts, so 1, 2. And then what happens here is we have what is known as the central aqueduct. So this right here, this spot between the third ventricle and this little tube that surrounds the spinal cord is the cerebral aqueduct all the way down to the central canal. So we have our cerebral, this is our cerebrum. So this means, I wish I could like make it bigger. Maybe I can. So I'm going to make this circle here, and then I'm going to show you what I mean. So here, this is our third ventricle, okay? This is our cerebral aqueduct. Okay, so remember, we have the interventricular foramen here. Okay, that's fine. Okay. Yes, I can still scroll. Okay. Okay. Back to our thing here. Just gotta plug in my iPad again. My apologies. Okay, so we have our lateral ventricle that's connected here. And so what we know is, oh dear. Perfect. Okay. You still see it scroll? Okay, perfect. So this is our lateral ventricle. This is our, so lat. This is also known as IV, so the interventricular foramen. I believe. Yes. So then this is our cerebral aqueduct right here in between the third ventricle. These are essentially, I'm going to explain what these even are. First I just need to show where everything is though. Cerebral aqueduct. Okay, so this then, the cerebral aqueduct then leads to the central canal, which surrounds the spinal cord. Okay. Okay. So that surrounds the central canal, and remember, this is all in 3D. So the central canal is surrounding your spinal cord all the way around. And the reason we have these ventricles at all is to regulate the flow of what is known as cerebrospinal fluid in order to keep your brain nice and insulated, and also keep it regulated, keep any materials that it needs, it keeps it in there, and then it also keeps it steady, and it makes it stronger, so it keeps it from being damaged. So then we have, let's see, the fourth ventricle, okay, and that's the letter aperture. But what we're doing here is we have also some cerebrospinal fluid that flows around the cerebellum. So if I were to go and erase this real quick. So this is your cerebellum right here, okay? And your fourth ventricle is leading to the central canal, yes, so your fourth ventricle is leading to your central canal, but it's also leading to get around your cerebellum. And then we also have our lateral aperture, which is an opening which leads back up to the lateral ventricle. Well, actually, it leads back up to your cordius plexus, which, remember here, now, what's all the function of all these parts? Now, this is where we start to get into it. It is in the fourth ventricle, or the lateral aperture inside of the fourth ventricle, so lateral aperture, that it moves back up all the way to the cordius plexus in order to be renewed, and that is essentially the flow of water. It's like water, but it's just fluid. So cerebrospinal fluid is going back up to the cordius plexus via the fourth ventricle. And then we can also see that the central canal also does this. It flows down the central canal and then back up the other side. So it is 3D, but it's also like kind of cut in half. And then, let's see, what else? Okay, so then how all of this works together, you may ask, because this is all very complicated, how it all flows back. Let's just draw a very big brain so that we can note it. So we have our brain. Let's just simplify the brain here and make it a big old circle, okay? What we can imagine is that this is our lateral ventricle, this is our third, this is our this is our central canal, our central canals, and then this is our cerebellum, okay? So fluid may flow through the cerebellum, like I said earlier, all around, but it can also flow starting through there and through the third ventricle. Actually, also through the central canal. So imagine this is also the central canal. This right here is the central canal. And the fluid is going to flow all around the brain in order to keep it protected and keep molecules flowing. So that's how it might work. And then you can see this growing, I can't. But the idea behind this is, okay, so not only do you have this place where it's flowing, but you also have this thing called the superior sagittal sinus. So this is where a lot of fluid will be flowing between your, and this kind of goes back to the meninges. It's going to be almost like two sections of your skull as a whole, and it allows for the flow of fluid. So we have these fluids flowing through. How do we get from the flow around the brain into the superior sagittal sinus? Now, note, right now we're in the subarachnoid space. So, okay, if you remember the meninges here, we have our subarachnoid space is like a bunch of webs. Okay, that's how you can also remember it's like a spider's web. And we have the cerebrospinal fluid flowing through there, but then we also have these things in here called the arachnoid granulations, okay? And these granulations can move cerebrospinal fluid into your superior sagittal sinus. And this is essentially going through the meninges of your brain, of going through, as we talked about earlier, your dura mater and your, below that, your subdura mater, and then your different parts. If I could scroll up for you, 1 second, I'm gonna fix this for myself again. Okay, so if you remember, we have our epidural space and then our dura mater, subdural space, arachnoid mater, subarachnoid mater. So what these granulations do is they go through all these parts of like your dura mater and such in order to get to your sagittal superior spinous. I don't know how to pronounce that again. Oh my goodness, again. I really just fixed it. Just go ahead. Okay, so superior sagittal sinus. Okay, good. And how they get there is through the arachnoid granulations. So everything's flowing through each ventricle, and you eventually get all the way to the end where it's recycled into the superior sagittal sinus through the arachnoid granulations. And that flows then back into your lateral ventricle. Okay, so we finally get back there, and we are almost done. So we only have these couple pages left, and we will be good with section 1. Okay, so we went through the ventricular system. Okay, so we are now on to the peripheral nervous system, which we can divide into 2 main sections here. So the first thing we'll need to know is that the peripheral nervous system, okay, this is mainly based on the idea of neural cells that are outside of the brain and the spinal cord. Okay, so there's your brain, there's your spinal cord, but we have all of these parts that are the nerves outside of your spinal cord that lead to other things. Now, the question may be, how do we attach these neurons to the nerves inside of the spinal cord and eventually to the brain? Well, we have these things called ganglia, or they are clusters of somas, or cell bodies, and what they do is they allow for the peripheral nervous system to talk with the central nervous system. Okay, so then we also need to know a few other things about the peripheral nervous system. The peripheral nervous system, unlike the central nervous system, is not protected by the blood-brain barrier. So remember astrocytes and how they formed the blood-brain barrier. We don't have that in the peripheral nervous system. So only the central nervous system gets the astrocytes. So that means, or not the astrocytes absolutely, but just the blood-brain barrier in general. So that means they can be more easily harmed. But with that comes another fun idea, is that the peripheral nervous system, it can regenerate. So, as we said earlier, it's something to note about most neurons is that neurons can't regenerate, and this is called a process of neurogenesis. However, this isn't the case with all neurons. So neurogenesis is usually not possible except for in the peripheral nervous system, where it's constantly being attacked. So like if I were to just like start banging on my hand real hard, or even like take a hammer and try and like smash some of my nerves, that can happen, and eventually I will be okay, because those are more likely to be able to generate or regenerate. So we then can talk about each particular section of peripheral nervous system. So now that we've gotten the difference out of the way, can talk about the somatic and the autonomic nervous systems. So let's start with the somatic nervous system. So I'm just going to break it up here. Well, no, we're probably going to be more like this. So this is the somatic. And then this is the autonomic. Okay. So here we can, let's just read a bit about it. So it's composed of axons leaving and entering the spinal cord that bring information to and from the tissues of your body. The nerves, axons, that are found above the spinal cord that bring motor commands to muscles of your head and neck or bring sensory input from those regions are also part of the somatic nervous system. This system is under voluntary control, meaning that you are conscious of sensations carried by the nerves, and you can consciously feel and see things from it. And this is different from other parts of the nervous system. So the main takeaway from this should be it's connected to your spinal cord, okay, and it's relaying information to the brain, such as with a muscle. So like if you have a muscle here, and that's your bicep, and you have your hand, can't draw a hand. That is going to be voluntary. So if you're moving your hand up and down like this, this is an example of your somatic nervous system. Now, on the other hand, we have our autonomic nervous system, which was actually one of the prompts on a practice essay you can do for science. So, in order to teach this, I'm actually just going to walk us through that prompt because it perfectly ties in to how our autonomic system works. So this is the other division. So if actually I were to do a little diagram here first. This is the breakdown. We have the somatic, and then we have the autonomic. Now, you see the word auto here, or the root word auto, meaning from Greek autonomia, or automania, I think. That's how you pronounce it. And that means independence. So auto, independence, and then this is voluntary. So I'm sure you can guess what this means. It means it's doing things on its own. It's not you thinking about it. So what's important about the autonomic system is it's broken into two parts. You have the peripheral, and then you also have the main two parts here are the two divisions, which are the sympathetic and parasympathetic. So, to illustrate these two different parts of the nervous system, I am going to pretend to be a friend, okay? Right now, I'm going to be the sympathetic friend. Because what I'm thinking here is, say I have a friend here. I'm Bobby, and this is Bob, okay? So I'm Bob, and I'm talking to Bobby, and I'm saying, hey, I'm really mad because, you know what? This beast over there, that bear, it's really scaring me, you know. And then the sympathetic Bobby is like, oh, yeah, I'm mad at that bear. Yeah, I better heighten my senses. I better be ready to fight. I better be ready to fight that bear. So, if you can't tell already, the sympathetic system, or division of our autonomic system, is the division of our bodies, which is our fight or flight response. It might be sympathetic and be like, I'm going to fight. Or, you know what, I'm just going to run because it's a bear. So, that is where the idea of the sympathetic chain comes from. So, in our body, we have our spinal cord, okay? And then let's say we have eyes, okay? And those eyes go to our brain and they tell it, hey, danger, okay? Danger, okay? We have this thing known as the sympathetic chain, which is a bunch of axons connected together that have ganglia on them, as we talked about earlier, that send out a signal to all these different parts of our body to tell it, hey, watch out, we need to be alert, we need to be ready to run or fight. So we have all these heightened senses. And this, down the long line of our spinal cord, is known as the sympathetic chain. So everything leaves really quickly and works well. And then we have different parts that are targeted, and it's effective, but it also takes a lot of energy. So since it's so fast, it's taking a lot of energy from your body, while the parasympathetic division is your rest and digest. Okay? So imagine your parasympathetic division like the evil twin, or even good twin, whichever you'd prefer, the twin brother of Bobby. And he's sitting there and he's thinking, like, he's sitting next to Bob, and Bob is saying, oh my goodness, there's nothing happening. And the parasympathetic division is like, nice, nothing's happening. You know what I'm gonna do? I'm just gonna start to count rocks. 1, 2. So, if you can't tell, this just means the parasympathetic division is gonna be doing work that is slower and more processing things in your body. So it's going to have things like immune responses, cell division and growth, digesting food, energy storage, and other aspects of homeostasis. So we have these two parts, okay? And what they each do is they act as this almost yin and yang between them. They work against each other because one is fast and one is happening when you're in danger and the other is happening when you're just relaxing. So we can think of it as this yin and yang here. Okay? And we have our comparison. I'm just going to use comparison. Okay, so these two parts, and that's where the prompt for the essay comes in. Because there, on the practice essay, there was this prompt asking, or just to compare, or not contrast. It was just contrast. Contrast the two systems of sympathetic and parasympathetic. And in this case, you may think, well, I mean, I don't know much about them. I can use details to talk about how they're contrasted, but what's important with these essays is if you want to try and do the best possible you can. You can't just, it is best if you just know all the information, like regurgitate these few paragraphs all about them. And also use this diagram here, which talks about like increasing heart rate, dilating pupils, constricting your, releasing your bronchi for your lungs to be able to breathe better, inhibits the secretion of hormones. Remember, hormones are slow acting, so that might be a waste of energy in these situations. And then we also have things like adrenaline and non-adrenaline, which we can release in order to create this need in your cells for more action. So there's all these responses. So you may be able to argue in one paragraph about all the responses of each, okay? So you may be able to say, like, in one, it's going to be, you can argue for just a few sentences and say, like, hey, the sympathetic is going to be your fight and flight response, as evidenced by constricting pupils, increased heart rate, not constricting, dilated pupils, increased heart rate, and the production of adrenaline. But you can also take it a step further, and you can say, then the parasympathetic division, by contrast, is more resting. It has a resting heart rate. So you can then say that this produces a yin and yang situation, and you can argue, instead of just contrasting them, you can say why it's important that they're contrasted. You always want to ask that to yourself when you're writing things, is ask yourself, why this, though? So, yin and yang is important to the body because it promotes, can you guess what word I'm going to say? Homeostasis. Okay? So, what you can then argue is say, hey, we're contrasting two different systems, the sympathetic and parasympathetic, and we're promoting homeostasis in the body because of those two contrasting systems. Okay, section 2. So we're going to get straight into how neurons communicate. So how this is going to work, it's going to be a little different than last section. We're going to read through together the section of writing, like between each heading, and then I'm going to explain it. So follow along as I go, and I'm going to skip through some sentences because they are unnecessary, but each section I'm going to read and then I'm going to explain it. So, neurons can send signals by releasing chemicals like many other types of cells do, but interestingly, they can also do something more unusual. They can send electrical signals very long distances across the body. Electrical signals sent by the nervous system are mediated by the flow of charged atoms across the neuron's cell membrane. Ions, which I explained earlier, are charged atoms which are negative or positive based on their electrons, can be found and move in and out of neurons to communicate through the body. The neuron might be in a particular state inside or outside the cell, which allows it to carry a signal. So these are known as membrane potentials, which is another way of describing the electrical charge that a cell has in comparison to the electrical charge found outside its membrane. So this allows for the conduction of signals. It's not just about, is there a certain electrical signal inside the neuron? No, it's about the inside and the outside that creates this charge. So these charges can be measured in millivolts, which are extremely small. And another important thing to note with neurons is that they have an on or off state. So we can look at a neuron. It's not going to be like slightly more conductive than another, or like it's not going to be more positive or more negative than another. So then from there we can take a look at the fact that it's constantly changing. Okay, so that all makes sense. I didn't need to illustrate anything for that. Hopefully that all made sense as I just talked about, that it's the outside and inside that make up a charge of a neuron. Now, how those charges actually become signals in the neuron, it starts with the resting membrane potential. So, there are three main factors, and this was actually one of the questions on one of the comprehensives we took, was what are the three main factors which influence the resting membrane potential, and that is the cytosol, which is, if you remember what an axon looks like, and let's draw an axon here, and then we have our, I'm not going to draw the myelin sheaths, I'm just going to draw the axon. This is going to be a very useful diagram later on, too. So, this, all of the stuff on the inside of the axon is known as cytosol. The outside is known as EF, or extracellular fluid. And then this is your neuronal membrane. So these are the three factors that influence the membrane potential. And this is important because all three of these have to work together. As I said earlier, an electric charge is based on the inside and the outside. And guess what? The neuronal membrane, it can influence those two things. Another thing to note is that cytosol and extracellular fluid are mostly water, but they have ions inside of them, and that's where the charge is going to come from. So there's also a name for what the axon is surrounded by. So we don't just have some random fatty tissue that's surrounding it. It has a specific name. It's known as the phospholipid bilayer membrane. Okay, so the phospholipid bilayer membrane, it actually has two layers, as I said, bilayer. It means that we have a specific type of fat, a phospholipid. Lipid means fat. Phospho, it means it's connected by phosphate. So we have these little two sections that connect together to make up the membrane. Now, what's important to realize about this is that it's selectively permeable. Selectively permeable. Okay? So, what this means is that only certain things can travel through the membrane. So, if we were to take a look at the membrane up close, it looks a little bit like this. Okay? So, and it's this chain of a bunch of phospholipids . So what this means is that we have these two sections which are broken up, and that's why it's a bilayer, is because it has two layers of phospholipids, which are fatty acids combined by the top, and it's selectively permeable, as I said earlier. So say we have a big ol' protein here that's like, comparatively, it's like super big to the membrane. It won't be able to fit into the membrane. That's also the same with ions even. If we have an ion, it's not going to absolutely be able to go all the way through here. Even if it's small enough, it may not be able to because, remember, ions, they have specific positive or negative charges, and that means that it either can't interact with the inside of here or can't interact with the outside, because it's the charges of each of these fats that actually allow it to go through. So that means we have a problem. We need a selectively permeable membrane, and we need ions to flow through it so that we can get a positive or negative charge in here, okay? So that means we need little channels, okay? They're like canals, channels, that allow our ions to go through our membrane. So let's take a look at some of the types of channels we can have. So how about we start? Yes, let's start here. So I'm gonna keep most of this here, and then we're gonna talk about channels. Okay, so what these channels are said to be are selectively permeable as well. So only certain ions can go through these channels. So they're like an opening, but they're like a gated opening. So there is a certain process for one of the types of transport, and that is these just these open channels, okay? So what we can say here is, say we have a sodium ion. So remember, this is just an atom with a positive charge. Let's say it wants to go through this channel. Well, if the channel allows sodium ions through, if it's a sodium channel, then it can go through, and it can go through the membrane. And this means it is diffusing through to another place. It means it's going from one place to another. That's all diffuse means, is it's moving from one location to another. The idea behind diffusion, though, is that you have a bunch of different sodium ions, okay? So you have, like, imagine all of these dots that I'm about to draw. Imagine all of these dots are sodium ions, okay? And they're all next to this place where there's no sodium ions, okay? What will happen is the sodium ions will diffuse into the membrane because there's less. And since ions are almost constantly moving in the extracellular fluid, or in the cytosol, it could be the opposite way, they're going to eventually make it their way through the channel. And eventually, they're going to move to make the amounts on each side more even. A way of also imagining this, which is common in bio classes, is that it's the movement of these molecules is described as a bunch of rocks falling down the hill. If you have a bunch of rocks right here, and they're stacked on top of one another, they're going to start moving down the hill. Okay? And as they start moving down the hill, they're going to start to pile up down on the bottom of the hill as well. That means eventually, as they roll down the hill, they're going to eventually even out to be the same exact amount on each side. So this is exactly how what is known as a concentration gradient is formed. So what a concentration gradient is concentration is the amount of a substance in a particular amount of space. And then a gradient is this triangle. It's the movement from one place to another. So what we say is happening here is it's moving molecules, ions, from one location to another because they're all in this location and they're so bunched up that they need to roll down the hill and go into the cytosol. Okay. So, yeah, and this is, that is the idea of concentrations. And now I will go set up the next part. Let's see. Okay, so next we're going to talk about electrical potentials. So how this will work is we have our concentration gradient. As I said earlier, they move in through a specific channel. But then what also happens with a relation of certain charged molecules? So if we have a bunch of positively charged ions, let's say sodium for instance, let's model them with a bunch of pluses, okay? And we only have a few pluses here. And we might have like more molecules on this side. We might have other random ones that might push the concentration gradient along. So the concentration can't even spell. Concentration. And then slash potential. And then there's the electrical gradient slash potential. Okay, so what this means is, say you have a bunch of positive ions together. You know that with magnets, for instance, we can have two sides of a magnet, a positive and a negative. They attract, okay? So they'll go towards each other, the positive and negative parts. But then if we were to take two positive magnets, they would repel from one another, right? So that means that in this instance, we're going to have a bunch of ions that are repelling from one another, which means they're more likely to want to go into a space where there's less pushback from them of positive ions. So this means that there's this other gradient, which is dictated by the electrical charge of ions as opposed to just the amount that's there. And this is going to be important to keep in mind when thinking about how neurons send signals. Okay, so let's redraw that because we're going to need it. Okay, so as I said earlier, there's these things known as ion channels because certain ions can't just be let through the membrane. So what we're going to do here is we're going to realize that, okay, we have ion channels that let certain things, and in this case we have inside of our neuromembrane, the neuronal membrane, we have potassium channels, okay? And these aren't any channel, they're known as potassium leak channels. And the reason they're known as this is because we have at all times in our bodies a buildup of potassium on this side of the neuronal membrane, inside of the cytosol, which we'll get into soon. But we have this buildup. So something to remember at all times is that we also have these leak channels that let potassium out. Since we have a high concentration of potassium ions, then they're going to be more likely to go outside of the cell where there's less of them. And in this case, there's more sodium ions on the outside. Now, why does this happen, you may ask? Well, originally, if we were at equilibrium potential, which means the charges and the the concentrations and the electrical charges of the different ions will be the same. So let's say we have like 3 sodium, 3 potassium, okay? And then on the other side, we also have 3 sodium and 3 potassium. Okay, so what our bodies do with this is in the axon, they have this potential. And we have a thing known as a pump, okay? So this is a very specific to different molecules. The pump we're going to be talking about in this case is known as the sodium-potassium pump. And I'm going to illustrate it in a little diagram, okay? So in normal equilibrium potential, we are steady here. If we were to just have channels, they would be equal like this. But let's say we had a different type of channel where it's only letting certain molecules through. So this is a pump, okay? This pump, I'm actually going to zoom in on it here so that you can see it better. What this pump does, and I can actually scroll down to an image of it for you as long as I get this working. Okay. Here is what is known as the sodium-potassium pump, and we'll come back to ligand-gated channels. But and voltage-gated channels. But what we can see here is if we had a membrane, okay, so let's make this a very big membrane, and then this is our cytosol. So remember, that's the inside, and then the extracellular fluid, EF, okay. If we were to have this, okay, let's illustrate here how this pump might work. So if we have 3 sodium, let's say we have equal amounts of sodium and potassium on each side. Okay, so we have 3 sodium here, then we also have 3 potassium here, and then we have 3 sodium here, and 3 potassium here. Okay, so you can imagine these pumps as like mini gates that are powered by energy. So what these also have is another little slot in them, and then we also have squares, let's do. Okay. So how this works is a bunch of sodium will come in. Let's model sodium by little circles.
1, 2, 3, and potassium with little squares. Okay, so just pretend we have like 3 and 3, 3, and 3. Okay, so these 3 are going up into the pump. Okay, so they kind of just wander into the pump, and this is usually pushed by forces such as the electrical potential and concentration gradient. Because another important thing to note is that in our membrane, it's not like this is constantly the same balance at all times. It's important to remember that even at equilibrium potential, this potassium could come over here, and then this potassium could move over here, and it's still at equilibrium potential because molecules are constantly moving. So it's important to realize that they're going to be moving back and forth between the membrane. They'll just be staying even most of the time. Okay, so back to the sodium-potassium pump. These sodium molecules move into the pump. And then what happens is a nice little friend over here called ATP, our very own energy, goes into this other little slot, which changes what is known as the conformation of the pump, which means it changes the shape. Now, you may ask, what are these pumps made of? They're made of proteins. And proteins have this special property where if they have anything added to them, they are going to end up changing shape and be able to do different functions, because we've learned that the morphology of given molecules or cells often relate to its function. And so it will change its conformation, okay, and it will then move. I'm going to erase this top part real quick. It will then move to open up to the other side, where the 3 sodium will be let out of the pump. All because we added this little ATP. We now have 3 extra sodium. So we originally had 3 sodium and 3 potassium in here. Now we have 6 sodium and 3 potassium on the outside. And this is also going to push some of the potassium into the pump. And how this potassium works is it has 2 slots. Okay? So in the pump, it has 2 slots. And 2 of the potassium are going to go into these slots. Now, there's not 3 slots, you'll notice. And this is on purpose, because now the ATP has kind of done its job, and it's now broken. So, it's now has the protein, the pump, is going to have these potassium in it now, and it's going to start to change back to its original shape. So if we go back here now, okay, remember we're keeping track. We have 3 on this side now, and then we have 1 and 6 on this side, and then we have 2 potassium inside the pump. And 2. Okay, and then we don't have any sodium carrying with us. These 2 potassium come in, and now we have 5 potassium on this side, 6 sodium on this side, and 1 potassium on this side. And then it's unbalanced, right? We are no longer at equilibrium potential, but this is what we want. Because notice, these are each a positive charge, okay? Okay, so this is, let's say this is about 5 positive, and then this is 7 positive. So relative to the outside, this is -2. This is a -2 charge relative to the outside. So this is all about just relative charges, not absolutely that we're moving negative ions inside of the cell. We're moving positive ions inside of the cell, but at different amounts, which changes both the concentration and electrical potential of the cell. And when this happens, when the sodium potassium pump is on, this means it is at resting potential. And this may seem counterintuitive because, well, we're moving stuff though. We're using the sodium potassium pump. We're creating a negative charge on the outside. Shouldn't that conduct a signal? No. Conducting a signal is going to be the change in that electrical charge. So if we go back here, and take a look at how this might work. Zoom back out. In a resting potential relative to the outside, the inside is going to have a negative charge and the outside is going to have a positive charge because of the sodium potassium pump. So we can look at how ligand gated channels work. We earlier discussed the idea of how the ATP molecule is going into the sodium potassium pump. That is known also as a ligand gated, not channel, but it's a pump. So let's just illustrate each of these types here. So we had our pump. We also have a voltage gated one. So let's do just a channel. Okay. Voltage gated channel. Channel, voltage gated channel, a ligand gated channel. And finally, we have our pump. Okay? So what a channel does, and there can be... Why was it just working and then now it's not working? That's okay now. So how a channel works is it's pretty much, you can imagine it as almost always just open. Okay? So it's letting in and out molecules, almost like just an open door. Okay? Then a voltage-gated channel, we'll go back to that. So this is just an open door. I think this analogy should help. This is an open door for people to walk in and out of. Okay. Yay. Okay. And then we also have our ligand-gated channel, which this is almost like a door with a key that goes along with it. So we need a little key slot and we need a key in order to open that ligand-gated channel. What ligand means is it's that key. That key is the ligand. Okay? So the ligand needs to fit into the channel in order to open it. Then we go to a voltage-gated channel, which we can imagine as a channel which has no lock, only a handle, but that door will only open if it has an electrical signal going to it. So like one of those electric doors, it's like that, where with the ligand-gated channel, you needed a key to open the door. But in a voltage-gated channel, you need like an electrical signal. So like say you need to press a button in order to open the door, or something like that. And then a pump is more like, it's as if you yourself, okay, say you're standing by a door, okay, and you brought a battery, okay, you need to make sure you bring a battery if you're ever going to go to a pump, and there's your door, and you plug in the battery, and at first you don't know what's really happening, but then you suddenly see a big old piston come at you, and like it pushes you all the way through the door, even though you didn't want to go through the door. It pushes you through the door, and then suddenly you also see two other people just like suddenly like be pushed past you, and they go through the door as well. That's what a pump is you're having that ATP energy take in a couple molecules and push them out, and then get other ones against the concentration gradient. If we were to go back to our image that we did earlier of the triangle and the rocks falling down the concentration gradient, this would be like pushing the stones back up the hill. It's using energy in order to move against the concentration gradient. And this is important, like I said earlier, in trying to create the ability to change a charge within the ion. So there we have the different types of channels. Okay, so then we need to also know the name of this type of transport. So say we have a pump. Okay, we have say the sodium potassium pump here. And we have our membrane, our neuronal membrane, and we have our ATP, okay? So there are two different types of transport. We have our pump, but then we also have our channels, okay? The main difference between these two, which I've talked about, is that the channel, it moves with the concentration gradient. So it is therefore known as passive transport, because it's just letting gravity do its thing, you know. It's not actually gravity doing these things, it's just, in the stone rolling analogy, it's letting gravity do its thing. It's letting it move down the hill. It's letting channel, even if it's like ligand gated, for example, the channel is still just open to however the molecules are moving. And remember, the molecules are always working towards this equilibrium potential where there's going to be equal charge and equal concentration on both sides. So if we just have a gate, it's going to do that anyways. But if we have a pump, okay, this is specific to the types of molecules, and it will push out certain ones, which means with a pump, we're going to be doing what's known, so this was passive. You might be able to guess what it's called. It's called active transport. It's where we're using this ATP energy, okay, in order to move against the gradient. We're using our energy, so this is you, and you're pushing the rock back up the hill because you want it to roll back down again. And to further this analogy, you might wonder, why would I want to do that? Why would I want to push the stone back up the hill? Well, you're either forced to in an internal punishment, or you do it because you want to gain energy. Say we have this rock here, and we want to break this wall, okay? The reason we might want to roll this stone back up the hill is because we want to break that wall right there. We want to break this brick wall. And we need energy in order to do that. So we're transferring our energy up the hill, so that then the stone can go down and transfer its energy into rolling down the hill and hitting the brick wall, so that it can open and we can get through it. So that's the idea behind active transport, is we're using ATP energy in order to move molecules across the gradient. And let's see if I missed anything. As I said earlier, it changes the shape or conformation of the pump. Another thing to note is this ATP that's being attached. It's not actually just an ATP. It's actually a part of the ATP. So the ATP is actually structured a bit like this. So you have the main part. Imagine it's like the A part, adenosine, which is like a sugar, connected to 3 phosphates, okay? Adenosine triphosphate. We have 3 phosphates. How ATP works is what it does is it cuts off one of the phosphorus molecules, it cuts off one of the phosphorus molecules, and how energy is stored in molecules is through its bonds. So, in detaching the phosphorus now contains some energy, and the phosphorus can then go all the way over to the pump, and it can attach to it. And it'll then change the conformation of the pump. Then the phosphorus will be, or the pump will be known as phosphorylized. So phosphorylized. I cannot spell. Okay, so it's phosphorylized. Goes to dephosphorylized. This means that it's, I'm going to make sure I use the right words. Dephosphorylized. Yes, dephosphorylized. Okay, so it's going to be dephosphorylized when the phosphorus moves off of the channel. Because remember, once the phosphorus attaches, the pump is going to look like this, and it's going to open to the other side to let the molecules out and the other molecules in. And then once the phosphorus leaves, it's going to change back to its original shape, right? So then it's dephosphorylized, and we can use it again. Yay us. Good job, body. Okay. So that's how that works. Okay. So now we've gotten through the ideas of different pumps and the concentration gradient. And this is all leading to something, right? Because we've already discussed that this means we have a resting potential and such. But if I go ahead and just fix my thing here. Okay, so hopefully you can see the change as I go here. Okay, so as I said earlier, we have in resting potential the sodium-potassium pumps, which are constantly working at our benefit to make the inside of an axon Remember, this is all taking place in an axon, in the myelinated axons. And these channels are actually happening in the nodes of the ranvier. So you can imagine we have our myelin, and then we have our myelin, and we have our myelin, and the nodes of the ranvier are where these signals are going in and out of the axon. And so we have our sodium-potassium pumps, which are pumping out sodium and in potassium. And it's changing the charge on the inside and outside. And so we also have one more type of channel that's taking a play that I mentioned earlier. The potassium leak channel, which allows different potassium ions to move through the channel. Think about it this way. If you're in a room crowded with people, okay, inside of here, or crowded with all just people that are exactly like you, okay, you might get really bored in that place where people are exactly like you, because you're like, I already know that. Oh, yeah, I understand all of that too. You aren't gaining any more information. So this is room A, where you're at, okay? Room A. And there's a bunch of, and then there's also room B. And there's a bunch of people that are exactly like you. Let's say they're called ones, okay? So there's a bunch of ones in here. But in this other room, there are a bunch of twos, okay? And there are still some also ones, so that's pretty nice too. Like, you can still be around people that like are exactly like you. Like, imagine if it was just a bunch of clones of you and then other people, okay? So yes, I know this analogy breaks down. We don't always want to just be with clones, but or it might be cool to be with clones of ourselves. But the idea is you don't want to just be with people of your own type. You can also be with people that are different from you. So in this room, you might want to move over to room B, even though there might be more people total and you might get a little claustrophobic. It's worth it because you're moving over to where the twos are. And so this is what happens in your neurons. The positively charged potassium moves to where the positively charged sodium and positively charged potassium is. Because remember, we have the sodium-potassium pump. So we still have some potassium left on this side, but it's mostly sodium. So a bunch of the potassium is going to be moving out of the cell, and that allows for us to get even more negative on the inside during our resting potential. Okay? But the thing is, this is, okay, equilibrium potential, we ain't at that yet. Okay, so what's important to realize is we have all this positive charge on the outside and not as much positive on the inside. So then we say that the inside of these axons, they are at about negative 60 millivolts as compared with the outside. So you need to keep in mind this is all in comparison with the, is that working? Yes. It's all in comparison with the outside of the cell. So this is negative 60 to negative 80 millivolts. So negative 60 to negative 80 millivolts, okay? And this is important to realize because we now have a charge inside of our axon, which means we're ready to send a different signal down that axon and eventually tell the other neuron, like, hey, this is my signal to you. And that's how neurons communicate, just as we talked about in the first section. So, before we move on to, and I'll even say what the name is, from resting to action potential, we need to understand another potential, and it is the equilibrium potential. Okay, we mentioned this earlier, okay? So what an equilibrium potential is when everything is at the same electrical potential and concentration potential. So, or concentration gradient and electrical gradient are at complete balance. That means that we have some positive ions and some negative ions, and we have the same amount on each side. So like, this is a good example. We have 3 negatives on each side and 3 positives. Notice how if I were to change, say, one of these on the inside to a positive, we would still have an equal concentration, an equilibrium concentration, or a concentration that is at equilibrium, but we would not have an equilibrium electrical potential. Because we have 4 positives and 3 positives, and 2 negatives versus 3 negatives. Versus in the equilibrium potential, we have, of each of them, we have equal amounts. Okay, so I explained that. Okay, then let's get into two types of equations. The Nernst equation and the Goldman. Goldman. Okay? Okay, so with the Nernst equation, both of these equations are just trying to find how permeable a cell is to certain ions. So, actually the equilibrium potential. So, the potential for it to move from one place to another. So, in the Nernst equation, we take into factors such as the charge of the ion, we take into account the temperature of the cell, and we also take into account the ratio of external and internal ions. So these are the 3 things that are taken into account. But then, with the Goldman equation, we take all of these same things as the Nernst equation, and we add on the permeability of the cell to certain ions. So, remember I mentioned earlier that a cell a neuron axon is selectively permeable, which means it will let certain molecules in and not let certain ones in or out. So, for instance, let's think about the door example again. If we want to figure out how many people are going to, or how likely it is for a person to move through a door, we can look at all these different factors. Let's say we're looking through the lens of a Nernst equation. Let's say that this door only only needs positive charges. It only needs positive people. It only wants, if the temperature in the room is greater than 90 degrees Fahrenheit, I'd rather work with Celsius. Let's do 40 degrees Celsius. That's really hot. Okay, 40 degrees Celsius, and let's say we do a ratio of external and internal, so we can take into account, how many people are in the room. If there's a greater, a person count of greater than, say, 10 in the room, then a person with these parameters can pass the door. Okay, so they're a positive person, they're a happy person, and it's greater than 40 degrees Celsius, and it's greater than 10 more people than the other side of the door. What the Goldman then adds is just the permeability to that specific type of ion. So it just looks at the person and it's like, are you fit to enter this door? If so, gladly come in. If not, no, you can't. And that's the idea behind it, is it's just a little more specific. Okay. And finally, we get to the action potential. Okay. I'm going to bring up the diagram for this because it's really helpful. I love it. Okay. So, remember, we are starting on our diagram with, now let's say this is 0. This is all electrical potential, okay? So, it's not all electrical, well, yes. It's electrical potential measured by the voltage in millivolts of the cell. So, let's say we have down here negative 60 millivolts, and this is around like negative 80. Okay, so this is negative 80. Actually, let's see. So we have just about negative 60 millivolts. Okay. So let's say our cell is chilling at 70 millivolts here, and suddenly something comes along, and it's like a knock at the door. It's like, knock knock, who's there? Action potential who? Action potential, I'm making you reach your threshold. So that brings us to negative 50 millivolts, because they knocked on the door. Now, our electrical potential in our membrane is increased to negative 55, which initiates our response. So if any other lower amount would have happened, nothing would have happened. It would have just stayed at negative. So we could have even had like a little jump and nothing would have happened. If they knocked on the door too quietly, essentially. If they went like... No, they need to knock on the door like this. Okay? So now that we've reached negative 55, that's known as the threshold. Okay, so this is the threshold. Okay? And now that we've reached that threshold, we can keep moving up. Past 0, which is our equilibrium potential, remember, because we're at equal amounts of positive ions on each side. So this is our equilibrium potential. This is our, let's see, equilibrium potential. And then we make it all the way to 40 millivolts, okay? So this is our peak, okay? So depolarization. Okay, now that we've started going, I'm going to also illustrate this at the exact same time with an axon. Okay, so we have our axon. Let's just illustrate it with our double membranes. Okay, So what we're imagining here is we have the potassium that's mostly all in here, almost no sodium in here. And then we have sodium in here, which all of these are positive. You need to always remember what our charges are. I'm actually going to lessen this amount. Let's say we have 3 potassium there, and then we have a bunch of sodium on the outside, and even some potassium. What's happening here, okay? Let's say this direction was our soma, okay? So our soma decided, let's introduce some more sodium into here. And actually, let's make this membrane, let's make it a little bit charged. Once that happens, certain things start to open. First of all, our sodium-potassium pumps just completely break down. Like, they are not okay. Like, they're just like, they just stop working, okay? That's all you need to know, is that the sodium-potassium pumps, they're kind of sad, because they don't get to do their job anymore. But this is what happens with equilibrium potential. This sodium right here, and creating a voltage in the membrane, is what allowed us to get to our threshold. Okay, from there, how we get past where we are during the stage of depolarization is we have, okay, voltage, yes. Yes. Okay.
Blocking different will.
Okay. So what happens at about,
I think it's 0. No, it's negative 55.
Perfect. Okay. So at negative 55 here, at the threshold, we have what's known as a voltage-gated sodium channel. So we talked about voltage-gated earlier, voltage-gated channels. Okay, this is a voltage-gated sodium. This means that sodium is being let in from the outside, which means, wait a minute, the more sodium we have in here and the less sodium we have out there, the more positive it's going to become. This means that it's going to make it all the way up to positive 40 or about positive 35 millivolts. And this is the state of depolarization, because remember, we were keeping during our resting phase our axon negative relative to the outside of the cell. But now that we're depolarizing, we're making it less negative, and therefore depolarizing, we are making it less negative or making it less charged, and then eventually making it positive. So at that top, that's known as the peak. Okay. And then, how about we read through how sodium channels work so that we can understand how the absolute refractory period works. So, if we have voltage-gated sodium channels, we know that there is a large electrical potential creating an electrical driving force which also pushes positive ions into the cell. This is because the extracellular fluid is more positive than the cytosol and thus repels the sodium ions, which are similarly attracted to the negatively charged cytosol. Thus, there are multiple forces acting on the sodium ions, drawing them into the cell.
such that when the threshold is reached and the voltage-gated sodium channels open, sodium rushes in the cell very quickly. We talked about this depolarization. Then it eventually peaks around 30 to 40 millivolts, at which time the voltage-gated sodium channels become blocked or inactivated. So as you can see in this chart on the top, we have inactive channels. So then this blocking mechanism is different than the closed conformation of the channel, because while a closed voltage-gated sodium channel can and will open if the membrane around it is depolarized to the threshold, an inactivated voltage-gated ion channel cannot open regardless of what the surrounding membrane potential is. So this is known as the absolute refractory period, because, okay, we've reached our peak. We've reached 40 millivolts, okay? We have reached age 25. That's our peak years. Or if you peaked in middle school, 12. We have reached the amount that is needed to start back to relay our signal. So what happens? We have our sodium gated channels. I'm going to do a little close-up here. Okay, we have the little chart on which you guys can see. We have like a little thing here, and then we also have a little circle here which you can deactivate it. Once it reaches 40 millivolts, it's going to block any sodium from coming in, and that inactive state will prevent it from being reactivated. Now, this is important because it needs to be able to become more positive, it needs to become more negative in order to reset the signal. That way, we can not just have a continual positive signal that keeps going in. Our cells would then not be able to communicate because we have this negative signal at first, and then we have a positive signal, but if the positive signal just keeps coming because we don't have this pause period to be able to reset back to a negative period, we aren't going to ever be able to switch between negative and positive. And it's not the fact that it's positive that it's able to relay a signal, it's the fact that it's changing from negative to positive. So we have this absolutely refractory period in order to help the cell reset and go back to its negative. Okay, so then we are going back down the peak. Okay, so we're now approaching our middle age. This is also known as hyperpolarization. Okay? And in hyperpolarization, we are able to move from 40 millivolts down. Let's say we get down to 0 at where equilibrium would be. So what's important about this is sodium channels have opened. They've let them in. Then they're inactive. Okay, during this period they are inactive. But now voltage-gated potassium channels at 0 millivolts start to open. And the potassium is like, woo! We don't need to be with all this sodium anymore. Yay! So they run because I don't really know. Why are potassium ions afraid of sodium ions? Anyways, sodium ions are now moving outside of the cell, which is making the cell more negative again. We move more down the roller coaster. And then we can remember, let's see, recall that there's a greater, yes.
very fun. Different way of looking at it. Okay, so during depolarization and hyperpolarization, there's different effluxes or influxes depending on what it is. So a sodium influx means sodium is coming from the outside to the inside of the axon. And let's just move this slightly over. Perfect. Okay. This is an influx because it's moving into the cell. Then we have an efflux of potassium. Okay? So this means that we are letting potassium out. Now, as I said earlier, potassium, they don't like sodium as much. But there's a reason. And they also don't like themselves because, look, we still have a buildup of potassium on the inside. We do have those potassium leak channels, but they aren't quite enough. It's almost like trying to have one door to let 1 million people out of a room. It's just not going to work. We need lots more doors. So what we're doing here is we now have this super positive, at the peak, we have this super positive potential. And so that means that it's going to start to go back down, trying to reach equilibrium again. But in the process, it lets too much out, and it goes below zero all the way down to negative 80, okay? So, or this is actually negative 70. So we go below where we even started because so much potassium has been let out in an efflux out of the axon. And so, okay. So, this leads to the question, there's this undershoot here, okay? It goes below negative 70 millivolts, and it takes a while to get kind of back up to negative 70 or even negative 60, which means it's going to take more work for it to get above the threshold. Again, this is all just a part of trying to maintain an ability to transmit a signal, because remember, it's the change from negative to positive and positive to negative which it's the flow from negative to positive which allows for a signal to be transmitted. Then, that means we need a greater amount, which means potassium is known as the delayed rectifier. Okay, delayed rectifier. So, what this means is, okay, delayed rectifier is essentially it's returning it to its job to potential at rest, which means it's going to eventually turn back onto the sodium-potassium pumps. But it's also so the potassium channels are slow to close back up, okay? In this case, that's why they're called the delayed rectifier, is because they do its job, but they do it slowly, which means the threshold is going to be harder to reach at this point, which means we are now in, not the absolute refractory period, but we are in the relative refractory period. Oh man. So remember, the absolute refractory period was when we had an influx or a where is it? Yes, it's the sodium channels. So when we had an influx of sodium and the channels became inactive, this was the... Wait, no, I don't think this was the refractory period yet. I don't know where the word was. Yes, it was. Okay. Remember, the absolute refractory period was essentially meaning that we could not add more positive. Now we need more positive in order for it to work, in order for the threshold to be reached again. And then we have our, yes, as I said, relative refractory period, where we are in a lower state, and it means it's harder to reach the threshold to get back up to the peak and cause another sodium influx and potassium efflux. Okay. Next, we're going to read through together the action potential conduction, because after explaining all of that, we're probably going to need to know what it is. So, I'm going to go ahead and erase all this. Okay. Then probably, after explaining all of the electrical charge related things, I'm not sure how much is left in this particular section, but it is getting darker, so the stream will almost be over, and maybe I'll do another one tomorrow. But okay, so let's read through action potential conduction, shall we? To transfer action potentials and the information they encode from place to place within the nervous system, action potentials must travel down the neuron's long axon very quickly. Within a neuron, the action potential signal is first initiated at the base of the soma in the beginning of the axon border, at a zone called the axon hillock. We talked about this earlier. From the point of initiation at the axon hillock, the action potential travels down the axon. To do so, a chain reaction begins in which the action potential is carried down the axon, or propagates, by repeating the entire sequence of events just outlined in our description. So I just outlined how an action potential happens. That keeps happening down the entire axon at the nodes of the Ranvier because it propagates down it. Almost like how a lit fuse on a firecracker burns down, lighting each subsequent section of fuse as it travels, the voltage-gated sodium and potassium channels are triggered to open at each subsequent section of the membrane as the signal travels down the axon. When sodium enters the cell during the action potential, it diffuses within the axon, triggering the next set of voltage-gated sodium channels by bringing the next section of the membrane up to threshold. So this is just saying each section, it's going to be brought up to threshold. Then, if this did not happen, the signal would degrade or decrease over the length of the axon as some of the ions carrying the signal would diffuse away. So think about this. It's selectively permeable membrane, yes, but some ions still may make it through. So, yeah. Okay, let's see. Now, it's also important to note that the propagation of a electrical charge down an axon, it only happens in one direction, just like a firecracker. And then the absolute refractory period, yes, we talked about that. Okay, so we need to know that trying to
conduct these signals over and over again, it takes a lot of energy, but it can happen, and it can travel down the axon, which I will illustrate here. So imagine we have like a wire type thing, except this wire is special because it needs to access places each time. So we have let's say we have a positive and then a negative and then a negative and then a negative, okay? We're trying to get this positive charge to move down the axon all the way, right? The reason why the myelin sheaths are here is so that it can carry the positive charge all the way across, because it's a good conductor. Then it will increase the threshold, or it will bring the action potential to the threshold and allow sodium in at this node of the Ranvier, which is that indent between myelin sheaths on the axon. And we can move then the signal positive into the axon of the neuron, which is carrying the signal, and it'll signal all the way to the end. And this entire process is also modeled here in this model, and conduction down this is known as saltatory conduction. So this comes from the Latin word saltere, meaning to jump, which makes sense. Think about Spanish. Saltar means to jump. So you can think about saltatory conduction. It's jumping from one node of the Ranvier to the next. Okay. So now that we have made it to the synapse, and it talks all about that. Let's see how much. Neurotransmitter. Okay. So we go all the way to 46, which means okay. So now that we've gotten through that entire section of just electrical potentials, we are going to take a break. We have 10 more pages, and we'll see. I don't think we're going to be able to finish this entire section in today's live stream, but we may come back soon.